Okay, let's dive into this. Welcome to This Explainer. Today, we're unraveling an absolutely wild medical mystery. We're going to connect three things that, at first glance, seem completely unrelated. A tuberculosis vaccine that's over a century old, a super obscure piece of cancer research from 1989, and the modern global crisis of COVID-19. I know, it sounds exactly like the plot of a sci-fi medical thriller, but this is real, peer-reviewed science, and the biological hacking involved here is just brilliant. So, our story starts with a really bizarre, real-world observation made by epidemiologists right in the thick of the pandemic. They noticed something strange. Certain countries were experiencing noticeably lower rates of severe COVID-19 infections and deaths. So, what did these countries actually have in common? Well, it wasn't the climate, and it wasn't just demographics. It turned out to be a universal neonatal vaccination policy for a completely different disease, tuberculosis, using the BCG vaccine. And the data was striking. Studies found that for every 10% increase in a country's BCG vaccination index, which is basically a measure of how widely the population is vaccinated with it, there was a 10.4% reduction in COVID-19 mortality. That's huge. And on the flip side, countries that didn't have universal BCG vaccination or started it way later saw much higher mortality rates. Those are massive stakes for a medical detective story, and it naturally begs the question, what exactly is this vaccine? To really grasp this mystery, we kind of need to understand our baseline tool. BCG, or Bacille Calmette-Guérin, is a live attenuated mycobacterial vaccine. Now, it's been used against tuberculosis for over 100 years, and it's still given to more than 130 million babies worldwide every single year. It is, without a doubt, one of the most widely used medical interventions in human history. But here is the complication. That brings us to Section 1, the immunotherapy problem. You might be wondering, if BCG boosts the immune system enough to fight off severe COVID-19, why couldn't doctors just give everyone a shot of it as a standalone cure? Well, the danger is that BCG causes a generalized, non-specific immune boost. Think of it like a shotgun blast. It wakes up the entire immune system, but in doing so, it can accidentally amplify suppressor cells that dampen the very response you need. Even worse, the cytokines released by this broad response can trigger a deadly cytokine storm, which is the exact thing that makes severe COVID-19 so dangerous in the first place. So what we really need isn't a shotgun. We need a sniper rifle. We need targeted immunity, a highly focused amplification of only the specific antiviral cells. Let's move to Section 2, PPD, the homing beacon. To figure out how to make that broad shotgun-style BCG response highly specific, we actually have to flash back to the year 1989. Researchers back then were trying to cure cancer, and they were facing this exact same hurdle. Enter PPD, or Purified Protein Derivative. This is basically a complex mixture of peptides derived from mycobacteria. And here is the absolute magic of PPD. If you inject it into a naive host, someone who has never had the BCG vaccine, it does absolutely nothing. No response at all. But if you inject it into a host that's already been primed with BCG, PPD suddenly acts as an incredibly powerful carrier molecule, triggering a massive, hyper-focused cellular reaction. To understand how researchers use this to their advantage, let's break down the complex immunology into a super simple analogy. Think of your immune system as a military operation. Your helper T cells, they're the generals. They assess the threat and call in reinforcements. Then you have the target, or effector T cells. These are your specific soldiers, the exact cells meant to fight the specific cancer or the COVID-19 virus. Now, PDPD acts as a homing beacon, or like a laser target painter. And finally, you have interleukin-2, or IL-2, which acts as the fuel or battle rations that actually allows those specific soldiers to multiply rapidly. Okay, Section 3, how carrier presentation works. So the crucial point is the brilliant hypothesis proposed by H.M. Saxena and his colleagues during that 1989 cancer research. It follows this really elegant, logical flow. Step 1, you prime the host with the BCG vaccine, essentially training their generals to aggressively recognize mycobacteria. Step 2, you take the specific soldier cells that are actively fighting the disease and physically attach that PPD homing beacon directly onto them. Step 3, when those soldiers return to the body, the generals immediately spot the PPD beacon and release their IL-2 fuel directly onto those specific soldiers and nowhere else. And this brilliantly illustrates what's happening on a microscopic level. You've got the helper T cell acting as our general, and the activated T cell functioning as our specific soldier. And right there between them, the PPD is acting as a physical bridge, linking the general directly to the specific soldier. This tiny connection ensures that the incredibly powerful, focused secretion of lymphokines goes precisely where it's needed, completely bypassing all the other cells. By forcing this intimate cell-to-cell interaction, PPD ensures the fuel is only given to the right cells. And why is this absolutely crucial? Because it completely prevents the clonal overgrowth of the wrong cells. It dodges the toxic side effects of blanket immunosuppression or those massive unguided immune responses. It's quite literally precision biological targeting. Which brings us to Section 4, the missing red herring. So, fast forward back to the year 2021. Dr. Saxena realized his 1989 cancer hypothesis was the exact missing piece needed for the COVID-19 mystery. Here's the exact prime boost strategy they proposed for COVID. You take a patient's own anti-COVID B and T lymphocytes, take them out, and activate them in a lab so they express MHC Class II molecules. You're essentially putting up display windows on their surface. Next, you pulse them with the PPD carrier to paint them with that homing beacon. Finally, you transfuse them right back into the patient, who has already been primed with the BCG vaccine, whether that was in childhood or just 2 weeks prior. The clinical goal here is just beautiful in its precision. The result is a high-magnitude, totally selective amplification of specific antiviral lymphocytes right inside the body. Because the immune system's heavy artillery is focused entirely on the specific cells fighting the virus, it can mount this overwhelming response to just crush SARS-CoV-2. It effectively fights the disease while completely dodging that deadly cytokine storm we talked about. As Dr. Saxena perfectly summed it up in his 2021 paper, thus, PPD may prove to be the red herring in the BCG therapy of COVID-19. By linking the PPD to the COVID antigen, scientists found a way to redirect a powerful, ancient, generalized immune response into a highly specific, modern weapon. You see, the BCG vaccine wasn't the direct cure, it was just the primer. PPD was the missing red herring that actually allowed us to target all that power. It's honestly just an incredible demonstration of how adaptable our immune system really is when we figure out how to speak its language. Which leaves us with a truly fascinating question to ponder as we wrap up this explainer. What other modern diseases could this 100-year-old vaccine and its clever red herring help us cure next?